Before you dive into your Chapter 6 assignment, I wanted to briefly walk through how to do replication and transcription and translation on your paper. So within the lectures, we talk about the molecular process, but then there's also this sort of general idea of how this actually works. And in your assignment, you are going to be doing something very similar here. So I have drawn here our parent DNA strands. So here we have a double-sided DNA backbone and then our base pair sequence. Now in a regular DNA backbone, you would have your sugar phosphate backbone and then you would have your bases paired across the center using hydrogen bonding. But I have pulled them apart so that we can do some replication here. Now remember that your DNA molecules are nucleotides, are complementary. That means that A always binds with T and C always binds with G. And we're going to use the template of each parent strand to produce the other half of a new strand. And once we separate our parental strands, copy both sides, we are going to end up with two identical molecules. So let's go ahead and copy our strands. Now our DNA molecule is anti-parallel. That means one strand runs in the 5' to 3' direction one way. The other strand runs 5' to 3' in the opposite direction. So keeping that in mind, if this strand has a 5' end here, the complementary strand is going to have a 3' end there. I'm not going to draw the 5' yet because we've got to write out all of our base pairs. On our other side, this strand is going to have the complementary and anti-parallel strand here. So the 5' end is going to be present there. So let's start copying our DNA. On this top strand, our first base is going to be an A paired with a T paired with an A G paired with a C. Now I'm just going to continue G, C, T, C, G, A, T, A, T, A, C, T, T, G, A, T, G, T, A. And now we're going to add that 5' on the end. So I just followed my parental strand all the way through and just looked for the complement there. So now on the other strand, let's make the complement. A, T, G, T, T, A, C, G, A, G, C, A, T, A, T, A, T, T, G, A, C, T, A, C, A, T, and then this end is 5' on the parent, so the daughter strand is going to be 3'. Now if you look at this molecule, if you compare the strands, they should be the same. Assuming I didn't make any typographical errors, the top strand on this molecule should be the same as the top strand on this molecule. Bottom strand here should be the same as the bottom strand here. And if they are, you know that you have copied your DNA molecule correctly. So on the top there, we have DNA replication. Let's move on to transcription and translation. So I have the same strand here, and we are going to now transcribe it. So we are going to use the DNA template strand in order to make our messenger RNA. Now the reason that we use the template strand is that when we make our messenger RNA, it's going to be nearly identical to that coding strand. And that coding strand is the one that has the information for the gene we're trying to express. Now, the process of transcription is almost identical to replication with two major differences. One, in messenger RNA, when we do transcription of messenger RNA, we're going to make a single-stranded molecule. The other difference is that we are going to incorporate a uracil because uracil is found in RNA instead of thymine. So let's make our messenger RNA from that template. Again, we have to consider that our strands are anti-parallel, so our strand is going to start at the 5' end, and then we're going to make our strand incorporating uracil instead. So everywhere we have a T, we're going to have an A. A now is going to bind with U. G, U, U, A. C, G, A. G, C, A. U, A, U. A, U, U. G, A. C, U, A, and then C, A, U. Now you might notice now that our strand, this messenger RNA, is the same as the coding strand, with the exception that everywhere there was a T, we have now incorporated a U. So let's look at the final step, the step of translation. So I have rewritten that messenger RNA code down here at the bottom just so we have some room for it. But this process of translation is going to take that messenger RNA and is going to turn it into an amino acid sequence. That amino acid sequence is then going to be folded and processed and turned into a functional protein. So in order to translate our messenger RNA code into amino acid code, we need our codon chart. So you can find this in your book, you can find this in the lab, or you can find this exact chart located with your assignment. So before I start writing, I'm going to just walk you through a couple, because as soon as I write, that picture is going to move back behind my browser here. So AUG. So first base A, second base U, third base G. So that is going to be our start codon. Now, when it is a start codon, it is also a methionine, which you might see elsewhere on the table. Methionine is a amino acid. You don't see it anywhere else on the table because it's only one. There's only one methionine. If it is a start methionine, it's actually modified just a little bit so that the cell knows that this is the start. So first base, UAG, is methionine. Second base, let me change color here, is UUA. UUA. So that's going to be leucine. Third amino acid is CGA. So CGA, that's this one here, that's arginine. So let's go back over here. Let's fill it in. So we had start. This is a methionine. Then we had leucine. Then we had arginine. Going back to the next one, GCA is alanine. UAU is tyrosine. You don't have to remember all of these names, I just happen to know them. AUU is isoleucine. GAA is glutamine. And CUA, leucine again. And then CAU is histidine. So now we have our amino acid sequence. One thing I wanted to point out over here, leucine, is actually kind of a special one. Leucine is actually coded by 6 amino acids. So that's why we have these very different sequences, UAA and CUA, both coding for leucine. Last thing I wanted to mention, just going over our assignment here, if we scroll down all the way to the bottom, you'll have first a few questions similar to what I asked, and then some more conceptual questions. Then in the bottom, we're going to introduce some mutations into our original DNA sequence, and we're going to look at what is actually happening. So original DNA sequence, this G, CA, etc., you are going to use that sequence to convert it into messenger RNA. Remember, complementary and incorporating those U's instead of T's. Then you are going to use that mRNA to produce your polypeptide using one of the codon charts. And then you're going to fill in, if there are any mutations present, what those types of mutations are. Now you can find out the different types of mutations by just scrolling back up. There's a list of the different mutations you might find and what they do. And then you are going to make an analogy. Now this analogy part is probably the most frustrating things for students, and I think it's because they overthink it. You're essentially going to think about your protein sequence as a sentence. So our normal DNA is going to be a sentence. The cat ate pie. And then we're going to introduce some changes to it. So let's look at this example here. We have introduced a mutation where we have changed the second nucleotide. We've changed it from a C to an A. So when we make our messenger RNA, you're going to notice that there's a little change. We've gone from CGU to CUG. Now, you are going to convert it again into your amino acid sequence. You're going to do transcription, and you're going to take note of what happens. You might have already noticed we have changed from arginine to leucine. Now, we've done that by making a point mutation, a single nucleotide change, and we have created a missense sequence. Now, a missense is where you introduce a different amino acid. So we're going to relate that to our sentence over here. So if we change the first word, which is essentially what we've done here when we've gone from arginine to leucine, by a single letter, in this case the second letter, we've changed the word here. So I have made the word make sense. I picked a word, a different word, where the T and the E from the original word are maintained, and I've only changed the second. I don't care if you actually make a word, because that's not the point. The point is that now this sentence doesn't make sense anymore. You have created a missense sentence by saying T cat ate pie instead of the cat ate pie. Do you see how we have ruined our protein there, our protein sentence? So I'm going to start or go through the next one with you. In this one, we make a change in the third base pair. So here we are going to be CGG, UCA, UGG, ACU. So if we pop back over to our codon chart, instead of the original CGU, we now have CGG. Oh, so here's something interesting. Now we have arginine, but we already had arginine over here. So we actually didn't make any changes. So we now have arginine, serine, tryptophan, threonine again. So we have the original sequence. So we have introduced a point mutation by changing one of our base pairs, but this time it's a silent one. It's one that doesn't actually change the polypeptide sequence. So I like to do, in this case, a slight change that when you hear the sentence, it doesn't change anything. So the change I make here is changing that E in the to an A, because when you hear it, the cat ate pie and the cat ate pie, you don't know which one you've made because that change is silent. Now I realize that on paper you can see that change, but you actually can see that change in this paper mRNA too, and that's okay. So you're going to go through the rest of these, make your changes, whatever is indicated, make your messenger RNA, translate it into a protein, look back and see what sequence change you've made. You're going to find at least 2 of them, sometimes 3, and then do the sentence as best you can.